Welcome to this lecture on cell biology, where we will explore the respiratory chain. Have you ever wondered why we need to breathe oxygen every single minute of our lives? The answer lies deep inside our cells, in a microscopic energy factory that powers everything we do. Today, we are going to uncover exactly how this incredible system works, step by step. Before we dive into the complex biology, let us build an intuitive understanding of what the respiratory chain actually does. Imagine a series of magnificent waterfalls cascading down a mountain. As the water falls from a high cliff to a lower one, it turns a series of water wheels to generate usable power. In your cells, the water is actually a stream of high-energy electrons, and the water wheels are tiny, intricate protein machines. As electrons are passed from one machine to the next, they lose a little bit of their energy at each step. The cell captures this falling energy to do something amazing. It pumps protons across a biological wall to build up pressure, much like pumping water behind a massive dam. Now that we have a conceptual picture, let us look at the formal biological definition. The respiratory chain, also known as the electron transport chain, is a series of protein complexes and electron carrier molecules embedded within the inner mitochondrial membrane. It consists of four main multi-protein structures named complex 1, 2, 3, and 4. Along with two mobile carriers called coenzyme Q and cytochrome C. These components work together to accept high-energy electrons from electron donors like NADH and FADH2, which are produced during earlier stages of cellular respiration. Finally, the electrons are handed off to molecular oxygen, which acts as the final electron acceptor and combines with protons to form water. Understanding the structure leads us to ask, why does this microscopic chain matter so much to our daily survival? Without the respiratory chain, our cells would only be able to extract a tiny fraction of the energy locked inside the food we eat, making complex human life completely impossible. It is the primary engine for generating the proton gradient needed to produce ATP, which is the universal energy currency of the cell. You can see its critical importance in real-life scenarios involving poisons like cyanide or carbon monoxide. These toxic substances bind directly to complex 4, instantly halting the flow of electrons, stopping ATP production, and causing rapid cellular death. To truly understand how the respiratory chain extracts energy, we need to look at the thermodynamics of electron transfer. This brings us to a crucial mathematical relationship that bridges the gap between electrical potential and chemical energy. The formula calculates the standard free energy change, represented by delta G standard prime. This tells us exactly how much usable energy is released. The variable n represents the exact number of electrons being transferred. The symbol F stands for Faraday's constant, which acts as a conversion factor between electrical and chemical energy. When the right side of the equation is negative, it means energy is successfully released for the cell to use. If the number of electrons increases, the total energy released also increases proportionally. Looking closely at our previous equation, we saw a term for the change in standard reduction potential. Let us define exactly how to calculate that. This formula measures the difference in electron affinity between the starting molecule and the final molecule. Delta E standard prime is the total electrical driving force pushing the electrons down the chain. EA represents the standard reduction potential of the final electron acceptor, which pulls the electrons. ED represents the standard reduction potential of the initial electron donor, which gives them up. By subtracting the donor's potential from the acceptor's potential, we find out how much more strongly the destination molecule pulls on the electrons. A larger positive difference means a stronger spontaneous drive for electrons to flow. Now that we have our formulas, let us see this with a concrete example. We want to calculate the total energy released when two electrons are passed from NADH all the way down the respiratory chain to molecular oxygen. We are given the reduction potentials for both NADH and oxygen, and we know Faraday's constant is 96.5 kilojoules per volt per mole. First, we calculate the change in reduction potential by subtracting negative 0.32 from positive 0.82, giving us 1.14 volts. Next, we substitute our values into the Gibbs free energy equation. Multiplying negative 2 electrons by Faraday's constant and our voltage difference gives us our final answer. The negative sign is absolutely crucial here. It tells us that 220 kilojoules per mole of energy is released into the cell to pump protons. This massive amount of energy we just calculated is used for one specific task: pumping protons. Let us look at the exact numbers. In the respiratory chain, complex 1 pumps 4 protons, complex 3 pumps 4 protons, and complex 4 pumps 2 protons per pair of electrons from NADH. However, the carrier FADH2 is different. It bypasses complex 1 entirely and delivers its electrons directly to complex 2, which pumps 0 protons. To turn this proton gradient into usable energy, it takes exactly 4 protons flowing back through ATP synthase to make one ATP molecule. The maximum ATP yield for a given electron carrier is known as the P/O ratio. Knowing the proton contributions of each complex, we can formulate the theoretical yield for NADH. The formula for Pn simply adds the protons pumped by each of the 3 stations that NADH interacts with: complex 1, complex 3, and complex 4. Pn represents the total number of protons pumped into the intermembrane space per NADH molecule. Once we have that total, we find An, which represents the total number of ATP molecules generated. We do this by dividing the total protons by 4, because it costs exactly 4 protons to manufacture a single ATP molecule. If a mutation caused a complex to pump fewer protons, the total ATP yield would drop significantly. Let us apply this simple arithmetic to find the exact yield for NADH. We are asked to find the total ATP molecules generated from one NADH molecule. First, we add the proton contributions from complex 1, 3, and 4. 4 plus 4 plus 2 gives us a total of 10 protons pumped across the membrane. Next, since it costs 4 protons to manufacture a single ATP molecule, we must divide our total proton count by 4. 10 divided by 4 gives us our final answer. This means we get a theoretical yield of 2.5 ATP molecules for every NADH that enters the respiratory chain. This high yield makes NADH a very valuable energy carrier. Now, let us look at the formula for our other electron carrier, FADH2. The structure is identical, but the inputs change. The variable PF represents the total number of protons pumped by the chain for one FADH2 molecule. Because electrons from FADH2 enter at complex 2, which does not pump any protons, the first term in our sum is 0. The electrons then proceed to complex 3 and complex 4, which pump 4 and 2 protons respectively. Just like before, AF represents the total number of ATP molecules generated, found by dividing the total protons by the cost of 1 ATP. Because it skips the first pump, FADH2 inherently carries less usable energy. Let us calculate the final yield for FADH2 to see exactly how it compares. We need to find the total ATP generated from one molecule. Adding the proton contributions from the specific complexes it interacts with gives us 0 plus 4 plus 2. This shows a total of only 6 protons pumped per FADH2 molecule. Dividing these 6 protons by the cost of 1 ATP, which is 4, gives us our final yield. The answer is 1.5 ATP molecules. This perfectly explains why FADH2 is considered a lower energy carrier compared to NADH. It simply does not push as many protons across the biological dam. Having calculated the energy and ATP yields, let us look at the entire system in action. In this diagram, you are seeing the inner mitochondrial membrane, separating the matrix at the bottom from the intermembrane space at the top. Notice the 4 large protein complexes embedded in the membrane. Follow the red arrows. You can see NADH delivering electrons to complex 1, while FADH2 delivers them to complex 2. The electrons are shuttled by coenzyme Q and cytochrome C all the way to complex 4, where oxygen catches them to form water. As this happens, look at the blue arrows pointing upward. These represent the protons being actively pumped across the membrane by complexes 1, 3, and 4, building up that crucial biological battery we discussed. We have covered a lot of ground today, so let us summarize the most important ideas. First, the respiratory chain is the ultimate energy harvesting machinery of the cell, safely releasing energy from electrons to pump protons across the mitochondrial membrane. Second, this resulting proton gradient acts as a biological battery, which is absolutely essential for synthesizing ATP, the energy currency of life. Third, we saw mathematically why NADH yields 2.5 ATP, while FADH2 yields only 1.5. Finally, remember that molecular oxygen sits at the very end of this chain to catch the falling electrons. Understanding this beautiful, highly coordinated molecular dance explains exactly why we must breathe oxygen every single second to stay alive. Great job following along with these complex concepts.